welcome back to Basic Science series. So today what we will be doing is using illustration technique and design some of the useful interactions in immune system. As you can see here, I have made DC, which is dendritic cell, interacting with different types of cells here, naive T cell, and you can see antigen is getting presented. So in this video, what I will be doing is designing this entire process, entire structure, and at the same time, I'll also explain few of the things regarding these receptors. We'll also try to understand what is the function of dendritic cell. We'll try to understand what is the function of cytotoxic T cells, and so on and so forth. So it's going to be a fun video where we will know how to design these structures, and you will see that what are the things that I'm using from these different tools. And last time I made the video, and where a lot of students they asked this question that if you can show the entire view so that we can see what kind of things you are doing. Also, the suggestion was that if I just fast forward the point where I'm struggling to design, or I can cut it down. Okay, so I thought, okay, I'll do that. So in this video, what you will see is I'm fast forwarding the regions where I'm just focused on designing, and I'll just keep the region where I'm explaining few things after I've designed the receptor. So this way it will be more productive for all of us. Another thing, if you want to also follow through, you can open out the PowerPoint presentation. This is just the background that I've designed, and I really like this background. This helps me design things better, and it's also like a page where you're designing, so it's much better than the white page. It's not comfortable to my eyes. So you can use another background which is a little bit dimmer. Do not use dark background, because then it will be very, very difficult for you to design the structure. So you can just use the a little bit darker shade of the I don't know, it's like a matte color or a little bit dusty yellow color. I don't know, right? You guys decide. All right, so without any delay, let's start designing the process. Let's start understanding the process and also understand these receptors. All right, let's begin the video. All right, so this time what we will be doing is designing the structure of dendritic cell and how it basically presents the antigen to CTLs, which are nothing but cytotoxic T cells. So we will be designing some of the receptors, we will be designing cells, and then hopefully you will like the illustration, and after the end of the video, you should be able to understand the process of antigen presentation as well at the same time. All right. So the topic is basically exogenous antigen activation. And let's see where dendritic cell is helping the T cell out. So for that, basically we need to design dendritic cell. And now to design the dendritic cell, first we need to basically know what are dendritic cells, why they are important, and then we'll discuss these things once the structure is done. So what I will do in this case is also I'm going to fast forward some of the part where I'm designing the structure, and if you want to follow through, you can also follow through. Just slow down the video at that particular point where I'm not talking, it's just making the structure. All right. All right, honestly, believe me, it's not that easy. What you're thinking in your mind, and you're also translating the same structure on the slide, especially when you're doing all those strokes, it becomes a little bit difficult. Let's see, I think what I'm trying to show here are the dendrites. So I don't know. You decide whether I've done a good job or not. So you can see I need to design the nucleus at the center. Okay, looks much better. So that is the dendritic cell. We need a nucleus at the center. Now, nucleus usually it's not recommended that you make a round circle. You need to give a little bit of realistic view. Not having it has that specific shape. It should be a little bit like maybe oval. Definitely not circular. And if you want to do few extra things, you can also do that. Oops, let me redo it. All right, so it depends on what are the preferences you have, but now it's important to design the structure. So next thing what I'm going to be adding is the antigen presenting cell. So let me design that. I can also explain dendritic cell. So basically it's a part of the innate immune system where these are the cells that encounter the pathogen and then present the antigens of those the molecules from the pathogen, which we call antigens, to the other immune cells. So this is dendritic cell right now. Now I will be designing the structure of one of the important component of the immune system. So it could be a virus infected cell, right? And that will interact with the dendritic cell. Antigen will be presented, and then a cross presentation of the antigen to the cytotoxic cell will happen. So here in this side, I'm thinking to design that, and on that side, I think probably we can design the cytotoxic T cell. Okay. All right. So now you can see I have made one structure, another cell that is trying to basically show the antigen. Antigen we have here is that triangle. Right to dendritic cell. One way is that, and on the other side now we will also show the antigens that are getting expressed to the T cells, naive T cells. So for that, what we need to do is basically we need cellular structure. So you can basically reuse these cells, just change the orientation, and then I need a lot of structures, surface structures. I need class 1 MHC molecules, CD8 receptors, CD3 receptors, probably also CD28 receptor. These are important. And then ligand for the CD28 is CD8 T and 86. So with that complex, then we can complete the antigen presentation to the naive T cells. Okay, we will not go into the details, but as you can see, this is how you can create the structures, which are like receptors as their ligands. Probably these are some of the receptors that you usually see, CD receptors. And let's label that this side so that you understood, right? I'll explain this in my another video. Here I'll show the interaction with the naive T cell. For that I need to change the color, so probably let's show this in bright pink color, or probably little bit lighter color. So usually try to use the themes which are not that contrasting. Okay, here what is going to happen now, the MHC class 1 molecule will present the antigen. Okay, I can make it closer later. So let me design the structure, and you will see one by one. I'll also tell you what are the receptors that I'm designing. All right. All right, so we have one interaction that is done with the naive T cell, so we can start labeling these things. So let's start with the first one. So this is your cytotoxic T cell, and you know we use Arial font for all the labels. So I think this time I'll use a little bit bigger size so it's clear. This is TC cell. It's a naive cytotoxic T cell. Okay, what next? So this blue colored structure is the MHC class 1. So class 1 MHC molecule. Okay, let's label that. if you touch this dot, so then they are connected, you will see. All right, so that's the MSC class 1 receptor here. Okay, let's make it a little bit darker so that we can see. All right, so this is DC. Let's label this dendritic cell. DC. You can also put that inside here. Saves a lot of space. Okay, so you have MSC class 1 molecules. What is next? That will be presented to the receptors on the naive cell. Next is you also need a co-receptor. Small co-receptor will be present here. Correct. So let's make it in this color. So probably a little bit oval in shape. Another we need is the CD8 receptor. So in this case, that will be connected with the MSC class 1 molecule. So depending upon how you want to make it, you can basically design these receptors. Let's color it as like this. Okay, looks better. So we need to label these. I'm not a fan of like high, as I already told you, high contrast colors. I usually use low contrast colors, so I'm gonna probably make it a little bit less darker. Okay, so this structure is also there, so we can label this. In this case, this is CD8 receptor. So CD8, cluster of differentiation 8 in caps. Make it a little bit smaller so that it can fit in here. Most of the struggle you will face is how to adjust all these structures in the given area that you have. So I think I'll place it here and then just drag the arrow here. That's the okay. I definitely need to disconnect these two. So chemistry class 1, this can go here like this. Okay, now I'm going to design the rest of the receptors that are there, and then we can discuss what are the important structures or what are the important points regarding this particular interaction. I'm just left out with one receptor and its ligand. So let me quickly design it. So I'm just going to use, let's not do that. Okay, so probably you have a structure like this. And that is interacting with another structure. Like this. It's a multi-domain structure, and then they have the connection with dendritic cell. So need to change the position like this. Fancy. Looks good though. What should I do in this case? Let's make this structure like main wall structure. Okay. No. Okay, there you go. All right. We need to put some colors. Maybe like this. Yeah, it's nice. For that, we can use we can also reuse some of the colors, but that will put some confusion. So I'm going to use another one. So what are these? These are CD28 and CD80, CD86. This is a CD28, and on that side is CD80-86. Okay. Last thing, we basically need to take the cells in front. So the receptors are like this, or we should do it reverse. I don't know. You decide. You can also do the same thing. I think I like the way that they are. They are on the surface, not embedded like this, right? So they are, most of the part is hidden. I don't like that. Let's do this. Okay, looks good. All right, so this is the dendritic cell, and we know dendritic cells, they are the immune cells that play an important role in initiation and regulation of immune response. They are found throughout the body, but particularly they are present in some of the important tissues like you have lungs, skins, and digestive system, and lot of the body part and organs, they contain the dendritic cells. And dendritic cells, they encounter the foreign substance like pathogen, tumor cells, and then what they do is basically break down those into smaller pieces and then do the presentation. Now, if we discuss the role of cytotoxic T cells, cytotoxic T cells are the T cells that are also known as CD8 positive receptors. So we have that CD8 receptor over here, and in case of helper T cells, you will have CD4 positive receptor. So that basically differentiates your cytotoxic cells from the helper T cells. And then you can see MSC class 1 molecule, its role is to present the antigen and activate the cell-mediated immune system. And there are so many, important roles associated with the cytotoxic T cells. We can say cytotoxic T cells, basically their role is to detect the abnormal cells and cause the destruction how they can do is they can basically detect those antigens via the dendritic cell. Here in this case what we have done is use the dendritic cell and showed the antigen to the cytotoxic T cells, and you have the T cell receptor over here, so that's the T cell receptor you have there, and then it uses perforins and gran enzymes that basically kill the infected cell. But here what is happening is that's why I told you it's a naive T cell. It's encountering for the first time. And there are other functions. It can also have the memory function where it will recognize the antigens or the pathogens, so that the response is again initiated if there is the encounter with the pathogen that happens again. Right? So as you can see, all these illustrations you can design by using simple software and simple tools in Microsoft PowerPoint, and you can make your presentations, whether you are preparing your report and you want to have diagrams, and you can see the quality of these diagrams are really fantastic, and they are publication quality images, let me tell you. So basically you can export these images, you can save them, and then you can use them to your final report and final paper, research article. Say you want to show the mechanism, the same way you can design all those things. So many things you can do. And I'll bring you more topics where we will interact. We'll try to understand the cellular pathways. We'll try to design some more receptors. So I will be designing a lot of different types of receptors. They are also useful for my research, and our group is working on various types of signaling mechanisms. So this is a useful way to generate these kind of structures. So now, since I have designed this structure, I can basically use this wherever I want, right? Whenever I want to design something like this, so I can take these small structures, and as basically it will be very simple for us to design these things if there is something else we try to show in our paper. So this is how you can use simple tool like PowerPoint to design things. I have already told you that you can also use Inkscape. That is also fun, but master one thing, and I think that will fulfill your all the requirements, because they have the drawing tools, and it's up to your imagination, what kind of thing you want to design. Look at this. I think I really I like this image for some reason. The color contrast and all the things are there. Receptors, they are interacting, looks good. Okay, so we have also discussed some of the things that are important. We have gone through the mechanisms, we have gone through the interaction. So interaction is happening over here. So there are various ways to interpret this interaction. Dendritic cell can also present the antigens to other cells can also present it's vice versa. So interaction is going on. A lot of different types of interaction is happening at the same time. And here we focused more on the T cell and dendritic cell interaction via MSc Class 1. So then I'll bring more videos on, and I'll be designing some of the important structures, some of the different types of structure in the area of cell biology, biochemistry, in the area of parasitology, molecular biology, and then sky is the limit, my dear friend. So if you want to see all those structures, and if you want to know how to design the structure that you can learn, if you want to at the same time learn and also understand the function, that is also equally, it's going to be equally useful to you. So please stay tuned, and definitely do not forget to subscribe to the channel. Till then, take care.